Hi, everybody. Welcome to another Chats with Chaudhry. I hope you're all safe and well. Today, I'm delighted to be joined by Bill Warren, who's product manager at Waters Corporation. And today, we're going to be talking about size-exclusion chromatography because Waters have launched some new columns in this space. So first of all, Bill, it's lovely to meet you. How are you?
I'm doing very well today, thank you.
Great. And where are you calling in from, Bill?
I'm calling in from actually Easley, South Carolina. I'm working remotely.
Fantastic. And I hope the weather's well where you are this morning in South Carolina.
Certainly.
Fantastic. All right. So before we start talking about size-exclusion chromatography, would you mind giving viewers who are not familiar with Waters Corporation a very brief overview of Waters and your own particular business unit and your own background as well?
That's a very good question. I've been with Waters for 36 years, and people still ask me, "Oh, you work for Waters? Do you analyze water?" And the answer is no, we don't analyze water. Probably 1953, Waters was founded by a gentleman whose name was James Waters. 5 employees working out of the basement of a police station in Natick, Massachusetts. And what he really remarkably put together was the first liquid chromatograph. It was used for the analysis of polymers. It was called a gel permeation chromatograph, and it consisted of some components from Dow Chemical, a refractive index detector, and a column, an injector, right, a pump, a data system. And back in those days, the data system was basically a strip chart recorder. And things have come a long way since then. Waters probably has maybe 7,500 employees, probably 35 working for 35 countries. And what's really interesting is that if you think about it, today's topic is on size-exclusion chromatography. Well, that's really a subset of gel permeation chromatography. It separates things based upon size in solution. So in essence, Waters really is very well known today as being not necessarily the father of HPLC, but really a leading manufacturer of columns, instrumentation for the separation of things that can be dissolved in liquids, thus liquid chromatography.
Fantastic. So let's talk about size-exclusion chromatography. For a layperson like me, what actually is it and how does it differ from the other types of chromatography out there?
Yeah, very, very good question. I think for those who are familiar with liquid chromatography, the separation is based upon several different chemical properties. It could be based on separation based upon the hydrophobicity. That's called reverse phase, ionic interactions, ion exchange. But this separation is based upon the size of the molecule in solution. That's just referred to as exclusion chromatography. Now, it's not the highest resolving technique. To separate something utilizing size-exclusion chromatography, things have to be roughly twofold difference in molecular weight. But what's really, really important about this technique is its ability to separate a monomeric protein from an aggregate. And as our viewers probably understand today, there are probably 150 monoclonal antibody therapeutics. And to have a product commercialized, this has to be safe and effective. And LC really helps answer those questions in terms of looking at whether that protein has aggregated or has fragmented. Within 10 minutes, you can provide that information to a customer, whether that customer is in the research stages of developing a therapeutic protein, developing that protein, formulating that protein, or manufacturing QC. So in essence, SCC separates things based upon size and solution. It is a gold standard, right, for its throughput, reliability to separate biotherapeutic proteins in particular. And today we're talking about monoclonal antibodies.
Right. Now, obviously, SEC has been there for a while. So what types of columns have people historically used to carry out SEC?
I mean, liquid chromatography goes back to the 1900s, and it basically separates things that were chlorophylls. But SEC was probably begun in maybe the late 50s, early 1960s, right? And the first separation medium was starch. It really wasn't a very good medium, right? So then it advanced to using cross-linked polymers, cross-linked dextrins. That was probably 1965. And the product that everybody used back in those days was something called Cefidex. It was a good product, You could separate things based upon size and solution, but it often took hours to do the separations because these polymers were soft. So you couldn't run them at a high flow rate, else they, right? So a real advent came probably in the 70s when people realized that they could use silica, which is basically sand, right? You can pour and enhance the pores within that sand. You could then separate things based upon size and solution, but much faster. You couldn't just use bare silica sand. They had to coat it with something. It was a diol coating, and that coating made the medium, the particles, more appropriate to separate proteins based upon size and solution, and more importantly, to minimize any non-desirable secondary interactions. Because silica has a lot of charges, and you don't want a separation based upon a mixed mode. You want a separation based upon just size and solution. So people have been using silica-based diol-coated columns and really have been able to do separations, we'll say, within a half hour.
Right. So if that's the case, why have Waters launched new columns into this space? And also, what makes them different to what's already out there?
You know, that's a real good question. Let me go back just maybe 15 years ago, because Waters recognized, as well as other companies, that really SEC was a gold standard for looking at therapeutic proteins, peptides, etc. Right? So Waters made an advancement 15 years ago by not taking just silica, but using a hybrid particle. It's called a bridged ethyl hybrid particle, and we put diol on it. So that was one of the advancements. And the second advancement was using a sub-2 micron particle. So now for the first time, you can get fast separations within 10 minutes, right? And less of these secondary interactions, right? Because again, the bridged ethyl hybrid had less of the silanos, the charges. So that really was a revolution or an evolution, all right, in terms of developing columns for this application area. So 15 years ago, our chemistries and other competitors, pretty good batch-to-batch reproducibility. Ours were very good. We test every column to make sure they are. Good resolution of things that are even less than twofold difference of molecular weight, but still some unmet needs. And those unmet needs were these undesirable secondary interactions, ionic and hydrophobic. So researchers had to spend a lot of time developing a method, looking at different pHs, looking at different ionic strengths, using additives like arginine to get a good separation. We recognized that 2 years ago, and our R&D people said, what can we do to overcome those existing challenges? So to your question, what we've done is we've developed not just a new particle, but a new column technology. So together, using max peak high performance surfaces on the hardware that minimizes the non-desirable ionic interaction, and taking the BEH particle, and this time using I'm going to get too technical here for our audience, but using a hydroxylated polyethylene oxide in combination, they synergistically minimize non-desirable secondary, ionic, and hydrophobic. Consequently, we have some examples, application notes, etc. People can use simple phosphate buffered saline and get good separations, not for all samples, but for many samples. That is a game changer because everybody wants to reduce method development time, cost per analysis, etc. To be able to get good separations in just buffer is remarkable. And that's what we're really excited to share. In addition, we also learned to make customers successful, they need to make sure they match the column ID and length to the LC system. And I don't want to talk about that today, but that's a very important characteristic that you need to understand to get good, reliable separations.
Right. Okay. So 2 questions. Are the columns available right now?
Yes. Second question?
Well, the second question I was going to say is that what do you see as the future development of columns going forward, or what the applications of these new columns, apart from, you just mentioned MABs as one of the applications for them, or what other applications could you see going forward for the new columns?
Yeah, I'm gonna share a few things with you. So the first question is, the column was introduced at the well-characterized biopharmaceutical meeting in Washington, D.C. on January 25th. So it's
Right.
been commercialized for 3 months. There are column sizes appropriate for any LC system. Don't want to get the details, 2.5, and also some for low dispersion systems, a 1.7, and a God column. What's the God column? That tries to, it will extend the column life. One of the major reasons of premature column failure is the injection of particulates, microbes. These buffers, PBS, will grow microbes. You leave it in the sun for a week or so, and you're going to look at it. It's going to be green. And customers don't appreciate that, all right? It's not usually that bad in the customer's lab. It's not green, but they're injecting microbes. So, after maybe 50 injections, the sample, the columns fail. So again, that was the new product we introduced. God column is matching to the analytical, minimizing secondary interactions, faster method development using PBS. Your second question, where are things going? Well, people always ask for faster separations, but there comes a point where I don't think you can go much faster than 5 minutes to get a good separation of aggregates, monomers, and fragments. So what remains to be done is looking at things that are beyond simple antibodies. I think we've all heard about viral vectors,
Sure.
vaccines, messenger RNA vaccines, lipid nanoparticles, right? So to do those, if you look at the size of those things in solution, they are bigger than most SEC columns, whose pore sizes go up to maybe 450, right? So an unmet need is developing a SEC column technology that is robust. You think about it, if the pores are really large, those particles aren't very stable. So that's a challenge. Big pores, big pore volume, stable particles to do challenging analyses, looking at aggregation of things like viral vectors, plasmids, supercoiled linear relaxed. And those are still some unmet needs that customers have, and it's growing because those are some of the future biotherapeutic solutions.
Fantastic. All right. Now, where can people get more information about the new columns and about SEC in general?
Yeah, sure. I would suggest that they go to www.waters.com/SEC. It's our landing page. And on that landing page, you will see a lot of information, videos, care and use manuals, which I want to get into what we do to try to help make customers successful, but there's comprehensive information there to help make customers successful so that they don't fail and then require help to make them succeed. We want them to succeed out of the chute. So that website's going to give you a lot of great information.
Brilliant, Bill. Well, look, Bill, I appreciate taking the time out to talk to me today. It's always interesting talking to you, and it's good to get a bit more detailed history of Waters as well. So thank you very much for taking time out to talk to me.
And I enjoyed the conversation as well. Sorry.
Fantastic. And viewers, there you go. If you'd like to know more about SEC and also about the new columns from Waters, then check out the link that Bill just mentioned. I'll put the address above the video so you can click on it directly. If you've got any questions for Bill, then leave them in the comments below the video. I'm sure I'll be happy to answer them. So all that's going to say is, Bill, thank you very much. Good luck with the new columns. I hope they go very, very well. And hopefully we'll catch up in person at a show sometime in the not too distant future.
So it's great. Stay well.
You too. And viewers, thank you very much for watching. Until next time, as always, stay well and stay safe. Bye-bye.